Hi, everyone. My name is Angela Wu. I'm so glad that you're still here. I know that in the afternoon you can get a little bit sleepy. I'm talking on the third part of our topic today, where we will discuss the ethics and also the sort of entrepreneurial applications and opportunities related to genome editing. And I have to thank Professor Gao and Professor Wang for giving such a good introduction. I really liked what Professor Choi said about learning the foundation first and then applying. So now you have the foundation on the science side, but when we innovate something, it's for humans, right? So you have to think about how it will impact society and how it will affect the people around you. So this part is where we will discuss that. Okay, so first, I want to wake you all up and take a poll. So on your thoughts, so far you have heard many applications. You might have read some things in the news about these new genome editing technologies. So what are your thoughts? Raise your hand if you feel that it is ethically acceptable to use genome editing on plants or crops. Raise your hand. Plants or crops. Okay, almost everyone in the room. I would say more than half. What about animals? Non-human animals. Okay, looks like fewer people. And then humans. Raise your hand. Much fewer people, definitely a minority. Okay, so I want you guys to keep that in mind, and then we'll discuss again at the end of the talk. All right, so as we've already kind of covered in the technical part, there are different types of genome editing. We have non-human genome editing and also human genome editing. So under non-human species, in the applications that the previous speakers have talked about, you can already see that there are sort of different spaces where these applications or these products will affect us. So we've talked about editing in lab animals to study health and disease. So we've done that in mice, in monkeys, and lots of other things, right? So this is mostly scientists in their labs. We just want to investigate what happens when we change a gene, right? We've also now have editing in microbes to help with production. So I'll tell you a little bit more about that. And there's also editing animals or plants, like as the previous speakers already talked about, for better crop production, better resistance, and so on. And some of these species will be released to the wild if we want to actually use them for what they were designed for, right? So you can think that you can consider that the potential impact will be much greater if we release this into the wild. Okay. So on the non-human side, then on the human side also, there is a distinction between what Professor Wang talked about in terms of ex vivo applications and in vivo applications. Meaning, do we do the editing outside the human body or do we do it directly in the human body? That's actually quite a big difference. And another distinction that I will make clear later is the difference between somatic cell editing and germline editing. Okay, so those are new terms, but we'll come to that later. So I just wanted to give you the overview of the different categories of genome editing. So far, a lot of times when we think about gene editing, we immediately think humans. We want to treat disease, we want to fix this and fix that for humans. And Stephanie talked about the fear of having designer babies. But I actually want to start with the non-human editing part. We don't talk enough about non-human editing. Here are some examples that might strike you. This is a picture of a macaque monkey, and it has been added with human genes that appear to inflict brain changes which seem to cause psychiatric disease. So these macaque monkeys, their embryos were edited with genes that human scientists thought or hypothesized that they might affect psychiatry. And then these embryos were put into mother monkeys and then given birth to baby monkeys. And these baby monkeys, after they were grown up, they show signs of depression and anxiety, right? So on the one hand, you can say this has really helped us to understand how primates, similar to humans, may develop psychiatric disorder. But on the other hand, is it humane for us to do this to these animals that are very intelligent and very similar to humans? Also, what really can we learn? We know that adding these genes in will cause these signs, but we don't understand the underlying mechanism of how it happens. So despite such a huge cost to the monkey, did we learn so much? The question is still kind of unanswered. Another thing is that, as you learned, the efficiency is not always 100%. So maybe we will treat 100 embryos and only 50 of them will carry the correct gene. And then when you implant those 50 embryos into mother monkeys, only some of those will come to term, but the mother monkey is still undergoing the suffering of labor, right? So we have to think about the sort of the humane treatment of animals in this kind of application. As I mentioned, there's also now applications to engineer yeast to produce drugs. So malaria is obviously a big problem, and artemisinin, recently awarded a Nobel Prize for the discovery to treat malaria of this drug. Artemisinin is from a plant, and these plants sometimes don't give a great yield of the drug. So now there are engineers who have put genes that are required into yeast so that the yeast can just be grown in a giant pot, and you can get drugs from that yeast. In addition to artemisinin for treating malaria, you can make yeast now that also create painkillers, like opioids, right? And these drugs are essential, definitely. But how do we regulate them to prevent people, bad people, from using these yeast for bad purposes? For example, opioids are also addiction forming, and people use them as recreational drugs, and this is not good. So we don't want bad people to create opioids using yeast very easily and then sell them and ruin the health of people, right? So how do we regulate this? On the plant side, you can create soybeans, for example, with higher yield and more resistant to climate change. But how do we promote equitable access? A rich country like the United States can have easy access to this technology, but what about the countries in Africa that actually really need it? Will there be equal access for all? And how will this impact the ecosystem? So a lot of these things, we haven't done the studies and we don't really know very well. Also, Alan mentioned the mosquitoes to using gene drive to try and prevent or reduce the incidence of malaria. He mentioned one method, which is to make the mosquito not able to bite. Currently, there are other methods used for gene drives that are already being applied in the world. One of them is to get the mosquito to produce a toxin that will kill itself. Okay? So this is, you put a poisonous gene into the mosquito, and the mosquito will make the poison that will kill themselves, right? So obviously, this will kill the mosquitoes that can carry the malaria, and then hopefully, eventually, you don't have mosquitoes and no malaria is transmitted. But again, does it pose a problem to the ecosystem? And also, what is the practicality of this? Because ultimately, this gene will be destroyed when all the mosquitoes are dead. And so but new mosquitoes may be coming in from other areas, and so we need to reapply this every year or every 2 years. So what's the cost, right? So all of these questions, I'm sure that not many of you have heard about being discussed in the press, but we really do need to discuss before we actually start to use them in the wild. So Alan also mentioned the editing of pigs. Another approach, rather than in addition to removing the viruses from the pigs to make the pig organ viable, is to make the pig cells more similar to human cells so that the organs can be directly transplanted into humans. What we call, in this case, is a chimeric pig with human or human-like organs that can be used for transplantation. But a very long, far-away ethical question might be, well, if a pig has a humanized organ, is it still a pig? What if we have a pig with everything else being a pig, but the brain comes from human tissue? I mean, it sounds kind of like science fiction, but hypothetically, right? Or even if it's not a human brain, let's say if half of the organs in the pig are human tissue, like heart, liver, kidney, those can be transplanted. Is that pig still a pig? Do we treat the pig like a pig? 50% maybe you think, it's okay. What about 60? What about 70? So these things are things that we don't think about so much today when we think about genome editing, but we should be talking about them. Okay, so to give you, just now, it was sort of just a big picture view of how it might impact humans, genome editing might impact humans. Let me give you a little case study to sort of show you how we can think about these problems and begin to discuss them to come to a good solution. So in the case of gene drive, Alan has already showed you that with gene drive, you add a gene into the mosquito, and when that mosquito mates with a mosquito in the wild, that gene will be passed on with very high efficiency so that in the end, all the mosquitoes carry that gene, okay? Now, a case study that really can apply to Hong Kong is the case of dengue fever. In 2018, just last year, around 20 cases of dengue fever were confirmed locally in Hong Kong, and it caused a lot of alarm. So you can see here there were newspaper articles saying fever outbreak, and you can see health officials spraying mosquito poison everywhere to try and remove the mosquitoes. So similar to malaria, dengue fever is spread by mosquitoes, a different species of mosquitoes, and it's a virus rather than a parasite like malaria. But in the severe cases, it can also cause death. Okay, so that's why Hong Kong officials were very concerned about this dengue fever. So there was some talk about using gene drive in Hong Kong to help eradicate mosquitoes and control dengue fever. My question to you is this a good idea to use gene drive in Hong Kong? If we look at the incidence of dengue around the world, like you can see in this map and in this chart, you can see the incidence for different countries in 2013 in the thousands right here. For Brazil, they have many millions, okay, of cases of dengue fever every year. Even our neighbor, Singapore, which has a similar size and population to us, they have 15,000 cases of dengue a year. In Hong Kong, we have 100. That's the order of magnitude that we're looking at for dengue fever, okay? Now, if we take Brazil as an example, where they have already applied gene drive in a few test cities, in Brazil, in 2016, they released these genome-edited mosquitoes into a few cities in the wild. The cost was over 8 million Hong Kong dollars for 2 years, and for deployment in one Brazilian city. The effects that they have observed so far is that there's no severe ecological effects so far, but the mosquito population in the cities was reduced by over 60%, and the number of dengue cases was also reduced by about 50%. So it seems like a very effective public health effort on Brazil's part, right? And so far, they're still doing these trials in their cities to see what will happen. Now, my question to you is, what about Hong Kong? So everyone has to make up their own opinion on this, but if we look at the benefit versus the cost, first of all, the economics, are we willing to invest this much money to address 100 cases of dengue per year? There are some long-term effects that we are still unaware of, right? What if the mosquitoes develop resistance? Then what next? We've already invested all this money. What do we do next? Or what if the gene drive jumps species? This has been known to happen in the past. Genes can spread from one species to another, although it's very, very rare. Okay? Do we have some approaches to sort of take care of this if this scenario occurs? Oops. And what if the dengue virus adapts and becomes spread by other animals? Then our gene drive in the mosquito no longer works. And the long-term effects on the ecosystem, again, we haven't studied that for long enough. And certainly there are other unpredictable factors. Humans cannot predict everything, right? So again, I'm not trying to scare people here. I'm not saying that genome editing is not good. Clearly in Brazil, it has helped millions of people to prevent dengue. But this is the case when we think about ethical questions. How do we think about the ethics of a technology that's so new and so complicated? I'm sorry that the color is a little bit weird, but basically we need to balance the risk and the benefit. So first, we have to identify what are the potential ethical questions. Then we have to list out all the facts that we know, and then think about what are the relevant perspectives in the ethical context that we care about, and who are all the stakeholders. So we need to balance the risk and the benefit. For Brazil, their potential benefit is much, much higher than the potential cost or harm that they risk by employing the genome editing. Whereas in Hong Kong, that benefit may not be really there, right? The other thing we need to take note of is we should not sacrifice a small group against their wishes or without their knowledge, even if you think it is for a greater good, right? So when we say, oh, is genome editing safe? Can we just try them on somebody who is sick? Well, it depends, right? Does that person willingly want to try it? So we have to think about the personal rights and their personal choices as well. And finally, we need to consult all the key stakeholders. So you, in this room, you will all be key stakeholders in the application of genome editing. We need to talk with scientists to get the facts. We need to talk about it with policymakers and lawyers and government officials to come up with the correct policy that takes into account not just the science, but also the ethics. So now, we've discussed a little bit about the non-human genome editing. Let's move to the human genome editing, which I know that many of you are excited about. So again, Alan and Bo have already told you that actually genome editing is not a brand new technology. It just emerged more prominently in recent years because of the development of CRISPR and the ease that it has provided us, right? CRISPR has made these applications develop so much more rapidly that we have been forced to think more quickly about these problems. Whereas in the past, these tools existed, they just were progressing slowly enough that we didn't feel so rushed, okay? So the second clarification I want to make is that there is a difference between genome editing that is heritable and genome editing that is not heritable. Okay? So the difference is that we have different types of cells in our bodies, right? We have skin cells, we have liver cells, we have blood cells. All of these cells have different functions, but only one type of cells will pass the genetic information from you to your future offspring. And those are what we call the germline cells or the germ cells. That would be the sperm and the egg, and once they connect, also the embryo, right, the zygotes and so forth. And if you change the DNA in the sperm or the egg of a person, then there's a good chance that that change will also be passed to the children, right? But if I only change the DNA in my liver, that will not affect my future generations at all. Does that make sense? So you can see that this is a huge difference in terms of the societal impact. If I edit in humans in an individual, it will not impact future generations unless I edit the eggs or the sperm, right? So this is a huge distinction that we should keep in mind when we discuss or when we say whether we agree that human genome editing should proceed or not proceed. If you look, again, the text is a little bit small, but if you look at the difference between somatic or non-heritable gene editing and the germline or heritable gene editing, the consequences and stakes are much different, right? If you pass on to your future children, then you will have to live with those consequences almost for all eternity if you edit the embryos, right? And so for most applications that we hear about today being commercialized, every single one of them are non-heritable genome editing cases, right? So from the sickle cell anemia treatment to treatment of macular degeneration to treatment of liver disease, all of those things are treating the somatic tissue, the somatic cells that will not be passed on to your children. The safety risks of genome editing will also vary greatly from application to application. So we've already talked about ex vivo editing, which is you edit outside the body, and in vivo editing, which is you introduce the CRISPR and the Cas directly into the body, right? So if you think about your blood cells, you can take them out. If I change them and they become cancerous, I can catch them in the dish. I can use all kinds of high-tech approaches to screen the cells that I've made and discard any bad ones and only re-inject the good ones into the patient. In that way, we can greatly minimize the risk of such a therapy to humans. But an in vivo treatment will be a little bit more risky. First of all, you don't know how to evaluate the off-target effects once it's inside the human, right? It's difficult to interrogate all the cells that are affected. We also currently don't have perfect ways of delivery. What we mean is when we give you an injection, whatever is in that injection will go all over your body. How do we control it so that it only goes to the tissue that we want? Like if we only want to treat the liver, can we make the genome editing tools only go to the liver? So there are lots of engineers who are developing and improving these tools, but they're not perfect at the moment. So the risk of having an unexpected side effect of in vivo editing is much higher. And this is why if you read the news and look at therapies using genome editing that are approved for clinical trials, almost all of them are ex vivo. There are very few, or much fewer, that are in vivo, simply due to the risk. So one example that Alan briefly touched on is CAR T, this chimeric antigen receptor T cell therapy, where you take out the patient's immune cell, the T cell, and you add or remove certain factors into that cell to cue that cell to go kill the cancers, right? So the example that Alan gave was some tumors, they're really bad and they will fake a signal and say, oh, I'm not a tumor, don't kill me. And the T cell will have a receiving end that will say, oh yes, I see this message, you are not a tumor, I will not kill you. So if we take the T cell outside the body and remove that receiver and put it back in the body, then the tumor cell can no longer fool the immune cell and you get better killing. So in this case, it's ex vivo editing, it's quite safe, and you can think about what are the alternatives for these patients. Many of these patients have a predicted lifetime remaining of a few months. So if you ask them, would you rather try this experimental therapy, which it could make you more sick, it could kill you right away, but the alternative is that you will die in 3 months. So in this case, the potential benefit to the patient is much, much higher than the potential risk, or that at least it's a risk that they are willing to bear. So many of the genome editing therapies that have been approved for trials today are those where patients do not have good alternatives. And ethically speaking, this is much more acceptable to most people. And so you can see for CAR T, there are multiple clinical trials going around the world against multiple types of cancer. And despite side effects and some mortality, because the patient's immune system responds too strongly and they might die, but the potential benefit is greater than the risk for most of these patients. So you can see this is just a few examples. If you're interested in reading more, you can look at Kite Pharma, which is a pharmaceutical company. They have done a couple of clinical trials in this area. And here is a press release where they showed that almost most of the patients, half of the patients are not only still alive after the treatment, but a third of them appear to be cured. There were, I think, 1 or 2 deaths in a bigger trial, but overall it was considered quite a good successful case. With treating sickle cell anemia, it's the same thing. You can treat the blood cells outside the body and put them in, and so the same benefit and risk sort of evaluation will go on there. All right, so now when we talk about weighing the benefit of risk and benefit, we have to consider what are the potential risks of doing germline editing versus somatic editing versus ex vivo versus in vivo editing, right? And then, of course, the question then for germline editing is, will it ever become mainstream and do we even need it? So one bioethicist has been quoted to say that if he thinks about all the diseases in the world that we could potentially cure with genome editing, fewer than 10, I think was the number, fewer than 10 are diseases that can only be fixed by editing the egg or the sperm. Almost all the other ones can also be treated by doing editing on somatic tissue, which will not affect your future generations. So the question of germline editing is very, very controversial, but in my opinion and the opinion of many ethicists, we don't need to do it because 99% of cases, we don't need to edit the egg or the sperm to affect the disease. And finally, of course, there's the question of disease versus enhancement, right? If now I'm saying, oh, I want to edit so that I can have more beautiful skin, or I want to have a taller height or a smarter brain, should that be allowed? Well, the ethics of that are not solely applying to genome editing. And in fact, this applies to many types of enhancements. For example, prosthetic arms or legs, even using a smartphone, right? Not everyone can afford to use a smartphone. A lot of the same ethical issues about equitability and social justice will apply there. So the discussion around genome editing should not be about whether we should allow it or not in somatic cells, but rather how do we regulate and how do we discuss the policy so that these sorts of Nazi-ish kind of futures do not come true, right, in the case of enhancements. Okay, so I will very quickly go through the commercialization aspect because I'm running out of time. Alan already mentioned that there are 2 major players in this space, UC Berkeley and MIT Broad, and you can see a ton of different companies have already been spun out. They both own intellectual property in the CRISPR space, and there's a lot of argument about who owns what and who should be allowed to do what. But this has not stopped other companies from inventing technology around it because, why? The benefit to the patients will be so high that no judge would ever say, you are not allowed to make a life-saving product because you don't have the intellectual property. So companies are creating technologies and products around CRISPR, even though the IP issues, intellectual property issues have not been resolved. But again, ethics and safety are very important. If you're not careful, you might end up like her. And the reason why I point out the story of Theranos is that when you have someone who has no experience working in biotech, they think they know what they're doing in tech. Tech and biotech are fundamentally very different spaces to be entrepreneuring in. I can give you a very quick comparison. If you look at the commercialization process of a software startup or tech compared to a medical therapeutic startup, first of all, the typical amount of money that you have to raise right in the beginning is hugely different. For software, typically we're looking at 1 to 10 million Hong Kong dollars as the first or earlier raises. For a medical startup, you would need 20 million to 100 million Hong Kong dollars to even start doing your R&D. If you look at timelines for software, I can have 2 computer scientists sitting in a room just coding away. within months or at most 1 to 2 years, there will be a product ready to launch. But with medical products, that's definitely not true. Usually it will be 1 to 2 years for the first milestone because you have to do animal safety before you go into humans, and then you have to do humans just to check safety, and then after that you have to check whether it's effective. So typically for developing these kinds of therapeutics or drugs, it will be about 10 years or even longer for a product to hit the market. In terms of talent or personnel that you need, for software, it can be a young, freshly graduated person. You can think about Facebook, right? Microsoft, all started by very young people. They had no experience when they started the company. So the typical CEO, CTO age for a software startup will be around 20 to 30 years old. This is very typical. But for a medical therapeutic company, you want someone, at least 1 or 2 experts in medicine or science with very deep experience. These people have PhDs, they have postdocs, they have probably a decade of experience working in a pharmaceutical company. So the typical age of the CEOs or founders of a medical therapeutics company, we're looking at roughly 40 years old. So this is a huge difference between the 2 types of startups, and that's why we shouldn't treat them the same. We shouldn't have the same expectation of how long it takes for a product to come to market for medical versus non-medical. And so you can just take a look at the examples of how much money was required. For CRISPR, Series A was 25 million, 43 million, and 15 million initial funding to begin with. But if you look at tech, WhatsApp, Dropbox, GoGoVan, you all know those. Their Series A was 8, 6, and 6.5 million. Their seed rounds were only a couple of $100,000 US. So you can see that if you are an investor, or if in the future you want to raise money for your own startup, consider that the difference is huge. And we shouldn't treat the 2 the same. And I think today, sometimes people make mistakes about how they perceive these 2 types of entrepreneurship. Okay, so just to sum up, I would like to say that in terms of ethics, extreme positions that reject or accept technology that has potential to change our lives is dangerous. We want to take a nuanced view and really think about all the different positions and perspectives. We want to do well-informed policymaking by exchange of thought between scientists, physicians, lawmakers, and advocates. And don't forget that ethics can be even specific to culture. So cross-national communication is also important. What we know about safety and what's ethically acceptable may evolve over time. In the past, some things were not ethically acceptable, like organ transplantation. Today, it is. But that doesn't mean that we should rush ahead to do everything, right? Again, be open-minded, use evidence, and consult key stakeholders, and be collaborative. Okay, so to end, now raise your hand if you think human genome editing is acceptable. Okay, I think I've won over a couple of people. All right, so thank you for your time today, and we'll be happy to take questions.